Okay, so I'm going to talk about data sharing. And I have no conflicts officially, although I have a very strong opinion about data sharing. So you could argue that's a conflict. But so I thought I'd start with a case story. It's a case I've told in the past. Actually, it was a case I told to a reporter and written up in The Atlantic as well. And that was over 6 years ago, my lab had detected a PTBN11 variant in a fetus with increased nuchal translucency. It's sort of the measurement of the back of the neck, seen on fetal ultrasound. And this variant had been observed in an Ashkenazi Jewish patient with Noonan syndrome and was reported as pathogenic by a very well-respected lab. I had tried to get access, this is before EXAC and Nomad and really any good population databases were available. And I had tried to get access to a data set I knew existed from Ashkenazi Jewish individuals collected in New York City to determine the allele frequency, and I was denied access. They just weren't ready to share that data yet. So with the evidence in hand, we reported the variant as likely pathogenic, and that couple terminated the pregnancy. And later, we found out that the variant had a very high frequency in the Ashkenazi Jewish population, in fact, in that exact data set that I was denied access to and was in fact benign. So that was a less than optimal outcome. The good news is 6 years later, we have resources like ClinVar, working with ClinGen, Nomad, produced here at the Broad with Dana MacArthur. And we actually have much better resources to think about this. And in fact, a very similar story happened a couple of years ago where I got contacted through email from a couple who was pregnant, and they had had prenatal genetic testing prompted by increased nuchal translucency, the same finding in the prior case. And that testing had revealed a variant of uncertain significance. And they were concerned, very concerned about their pregnancy. And they contacted me based on our ClinVar entry, which at the time was shown here, where we listed the variant as uncertain significance. GeneDx at the time also had an entry, also of uncertain significance. So all three laboratories were in agreement on the significance of the variant at that point in time. But she was just asking, is there any other information you have? And so, you know, I reached out. I both dug into our data to figure out what case this was found in, what the phenotype was. It was also another, I think it was a newborn case. And the patient didn't have great matches to Noonan syndrome. And then I reached out to GeneDx, and they actually had several cases. And the variant had been found in a bunch of exome cases tested for other reasons. And eventually, we aggregated all of the data together and were able to reclassify the variant as likely benign. And this is now the new entry in ClinVar with the reclassified variant. And the updated ClinVar entry now has both GeneDx and LMM as classifying the variant as likely benign. And the good news is that the couple continued the pregnancy and had a healthy baby boy in the fall of 2017. So that was a much better outcome. And why did that happen? It's because we shared our data in ClinVar, and they had a place to go to find information, right? So I would argue for the best outcomes for patients, we need to share data on a global scale and also critically evaluate evidence as a collective community. So I put together what I sort of think about as different types of databases that are in the public and accessible in some way to support data sharing. I've grouped them by what I consider primarily gene-level databases, so things like OMIM. Most of ClinGen's own knowledge base is focused on genes, whereas we send our variants into ClinVar. The G2 gene to phenotype database in the UK, the Genomics England panel app, I'll talk about those later, Orphanet, really more of an ontology, and the NIH-supported genetics home reference. These are all databases that mostly focus on classifying gene-disease relationships. And then there's variant-level data. These are interpretations. They could be functional annotations, allele frequencies, things like ClinVar, the human gene mutation database, which is proprietary, so you have to pay money to get access to it, LOVD. which has a lot of locus-specific databases, and then, of course, EXEC and now Nomad as aggregate allele frequency databases, but also with increasing annotations in there. And then there's what I would consider case-level data. These are individual patient data. And in the US, the primary database to date has been dbGaP for NIH-funded studies. In Europe, they have an equivalent, EGA, the, what is it? Genome Phenome Archive. It's actually EPA, or EPA. And then there's databases like Decipher, also in the UK, or LOVD, where they have individual patient entries. The UK Biobank, the All of Us Research Program. I'll talk a bit more about some of these platforms. These are really individual patients and any variants that they have and so on. So those are the types of databases. So I'm going to start and kind of go through these different types of databases and how we think about sharing data using them. So why would we share, Marina gave a long lecture about how we classify variants, but why would we share this data with each other? Well, hopefully we could actually standardize around the genes that we include in clinical tests. And this is just a figure showing the different number of genes in hypertrophic cardiomyopathy tests across 14 different labs. So why are they all different? If one patient happens to get a test at one lab, they might get a different result than the patient going to a different lab, and that really shouldn't exist. And we use this information to think about different types of tests and which genes should go in it, and ideally we could harmonize the standards and the classifications so that we could have consistent resources being provided both for research and clinical care. And here's some applications of these gene-level databases. So Ambry using a classification system for how they apply this in exome reanalysis. The Genomics England panel app, they define all their disease panels. Australian Genomics has also implemented that. Lumina has used gene curation to define their predisposition screen. The group at Children's Rady has used that approach. So all different groups using gene-level resources to guide their clinical and research applications. And this is a screenshot of ClinGen's knowledge base where you can go in, and I think you've done this now in the workshop, be able to search on genes and get any of the levels of gene-level classification, validity, as Marina talked about, but also actionability and dosage sensitivity. And I think you saw examples of this data from Marina around the different levels of curation that have been done. So ClinGen has done about 500 genes to date. Now, in terms of sharing this data, so it's one thing for ClinGen expert panels to go in and do all this curation, but as you probably saw, there's judgment involved in deciding how to evaluate evidence and come up with which classification. So depending on how many points you awarded for each piece of evidence and what you considered good evidence, you can arrive at different classifications. And in fact, we were working collaboratively or communicating with Naz Rahman's effort, where she was trying to make a gene disease map of the entire genome, and classified all these genes as either yes, involved in human disease, no, or maybe, and some were leaning this way or that way using a color scheme. So we actually did a comparison between ClinGen and the Raman gene disease map. And you can see, for here, definitive and strong, the green stuff is lining up mostly with the red genes, but you can see there's some differences there in how we're classifying, and there's differences found in every category. So clearly, we may be saying one thing in ClinGen, and her team is saying something else. So clearly, not both of us are right. So this is an opportunity to identify these differences through sharing our data, and then we can together work through why we're different and maybe correct some of these differences. So in some of the reasons for discordance are we may define diseases. In fact, we discovered that they were defining disease very strictly as high penetrance disease. It would not consider anything that was a low penetrance effect. So like CHEK2 would not be a cancer gene, that kind of thing. Also the purpose of curation, we're also comparing with Panel App, and their curation is around should the gene be in this panel, And sometimes that's based on phenotypic decisions, not validity. So why are you doing the curation? How are you using it is the basis for some differences. Sometimes ClinGen has curated it for one disease, but not yet curated it for actually the primary disease, and we're working to get that up, but that was the basis for some differences. And then certainly the date of evidence evaluation. If you did it 3 years ago and there's new data since then, it may be different. So we started diving into these differences, and really understanding them we realized it's hard to compare if you're using red and we're using definitive, do they mean the same thing? So we got all of the groups working in this space together under the auspices of the Gene Curation Consortium, and agreed to try to harmonize all of our terminologies and definitions so that we could actually collaborate more effectively in this space. We put together all of the terms all of the groups were using to classify variants. So you learned already from Marina ClinGen's terminology and system, but we lined up across all of these different resources, and you see they're all different terms. Color systems, different names, confirm, possible, probable. Even on the color systems, they were using the colors for opposite meanings in 2 different systems. So red was definitively involved in disease here, but red was not involved in disease in that system. So you can see how this can go awry quickly. So we agreed that we would work to harmonize this whole thing and clarify the overlap between our efforts, understand what each one was trying to do, and develop consistent terminology for both validity as well as inheritance, allelic requirement, mechanism, disease. And that would ultimately enable us to collaborate on gene curation projects. So we went through over the last year a series of Delphi surveys with both across our consortia as well as then launching into the public. And we ended up just finishing this and arriving at a set of agreed upon harmonized terminologies that each of the groups are going to, most of them are going to launch into production or at least map to. So that's a terrific outcome, and it will make this whole process much easier. Also, not only have we now harmonized around these validity terms, but we're also, so I'm involved in the Global Alliance for Genomics and Health, and we have a national initiatives program where we bring lots of countries from around the world that are implementing genomic medicine programs, either in clinical medicine and/or research, and we are starting to share resources. And one of those is actually this panel app that the Genomics England project has developed to define which genes are relevant for different indications, and it allows you to much more easily scale, particularly when you're in a country that actually organizes across the country, which is not quite true in the US. But it's a little easier for countries that have national healthcare systems. So they've launched this to support England, and now Australia is launching an instance of Panel App, and we're starting to push this out as likely a toolkit for nations to actually track this for which genes are appropriate to test for which indications. So that's an evolving project. So that's kind of where we are with gene level sharing, and hopefully over time we will really begin to harmonize how we use this information in clinical practice. The next area is really talking about variant databases and patient level data. So I think about 2 different models for, particularly as we get into really granular data. One are centralized databases where everybody submits into a single physical database. And then there's federated networks where everybody's data sits where it's generated, but you use application programming interfaces to communicate around the world to be able to share data, query, et cetera. But you never have to transfer the data. So these models are, this has been the traditional model, and ClinVar is an example of a centralized database. Everybody submits their data to it. It sits on the servers at NIH. dbGaP is also a centralized database. You submit into it. However, we have now emerging platforms, particularly as we try to share globally. There's a lot of countries that will not legally allow their data to go outside of their country if it's patient level data. And so you just don't have this ability to say, hey, Germany, can you send all your data into NIH's dbGaP system? No, not going to happen. So how do we think about scaling genomic analyses when we live in a world where we can't bring the data all together? So we have to be more creative, and this notion of federation, where we link databases and can exchange information, subsets of information, or answers, or aggregate results through that is a new way of really thinking about this. So here's some of the different submissions into ClinVar. Matchmaker Exchange is probably one of the first excellent examples of a very productive federated network for sharing data. Whereas Nomad is really a centralized database. So the reason Nomad exists is because it's generated off of all of the data that's been actually sequenced here at the Broad. And so it's all centralized, and therefore you can create all sorts of annotations, understand the quality of all that data, because the Broad has access to all of it, or Daniel's team. So ClinVar, as a centralized database, as I said, everyone's submitting into it. That has the advantage that it's much easier for us to, as I call, share and compare. So patient-level data, you're not often comparing one patient to another. But in variant interpretation, that's absolutely a critical part. So by centralizing that type of data, we can easily, quickly see where we differ in interpretations. So I would argue that variant interpretation is a great example where it's useful to centralize, versus patient-level data is where it's not that advantageous, except in generating aggregate stats like is done in Nomad. Although I think we can even, going forward in the future, as we better harmonize the underlying data structures, be able to create resources like Nomad even from federated data sets. So this is just a map of where all of the data that goes into ClinVar is coming from. So it's not just the US, even though it sits in the US, it's a global resource. There are submitters from 69 countries now submitting, with over 500,000 unique variants that exist with interpretations in ClinVar, and over 1,200 submitters to date. So why should people share variant-level data? Well, we've put together a list of reasons. Certainly, it improves variant classification. Actually, as a clinical lab director, I have now discovered that it's actually the easiest way to provide updates on the reports that you've issued for patients. The patient comes back and the physician wants to call up the lab and go like, my patient's coming in this afternoon. What's the current interpretation that you have of this variant? And I'm like, I'm teaching at the Broad right now. Can't answer that one. So I can just say, they now know to just go to ClinVar and look it up. So it's it really saves us resources in that manner. It certainly adds value through standardization and quality control. When we submit to ClinVar, if any of our nomenclature is wrong, they tell us that. There's all sorts of standardization that happens through the submission to ClinVar process. There's actually a lot of labs that use this as publicity for their lab, that they embrace data sharing. Certainly, it's now consistent with evolving medical and regulatory standards, and there's policies out from the AMA, NSGC, ACMG, that are all saying that this is important. AMP is actually working on this as well right now. And there's even labs that have really embraced data sharing as a business strategy. Now, to recognize the labs that are voluntarily sharing their data, ClinGen launched what I call the lab list. And we have defined a set of requirements that you have to meet to say that you are meeting these minimum requirements for data sharing to support quality assurance. And once you meet them, you get on the list as meeting those requirements. Now, we have been giving extra badges for good work beyond that. So you see some groups have badges there as well. And over time, some of these badge requirements will turn into required requirements. But part of this is moving from the carrot to the stick approach, which is now we have a list to provide to clinical labs and hospitals, healthcare providers, and insurers who wish to only order from or reimburse labs who meet these data sharing requirements. And so we've been working with payers who are now starting to put this into their requirements for who they will reimburse. interestingly, when we launched this in July of 2017, all of a sudden several major clinical labs all of a sudden got on board, Quest, LabCorp, and ARUP. So that's excellent. It's making a difference. But not all the labs are there yet. The other challenge, and this has been, we've addressed it mostly in the US, because some labs will be like, I can't submit to ClinVar, my patients haven't consented. So we wrote up, and this is Danielle, there, the first author, points to consider and really articulated that submission to ClinVar is variant level data. It is your opinion on a variant. It is not patient data. You do not need consent. And we wrote that out with involvement of both NIH in there as well to make sure that this policy was very clear and that clinical labs could point to it when their lawyers said, no, 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 we can't submit to ClinVar, it's patient data, and we can say, no, 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 that's not true. So that came out. Actually, the UK was struggling with this whole issue as well, and they ultimately came out with a very similar document to say, you know, based on the standards in Europe and the UK, that we consider this variant level data does not require consent, so on and so on. And that has been really helping the UK be able to share data as well. So critical things. And we also, on the ClinGen website, have videos and information around, because what we don't want to, we do consent patients to share extra data. And if they don't consent to that, we don't want them to think their variant's not going to show up in ClinVar. So we have all sorts of consent forms that explain, well, what's the difference if you don't consent versus what happens when you do? And so we sort of picture, well, when you don't consent, we just give aggregate information to support that variant's role in disease. But if you do give your specific consent, then we can give much more granular detail, like this variant was found in a female age 50 who was African American, had breast cancer, or that variant was found in a patient who had both breast cancer and developmental delay. And this granular level data can be really critical when you're trying to interpret variants. And so we encourage this detailed level, but there is a clear difference. No consent required, consent required. And that's all shown in these videos and now multiple languages. And so this is an example where we've submitted a single variant where all of the evidence is aggregated. It's been reported in 6 individuals with HCM. Now, we do sometimes give details, one with DCM, one with myopathy, one with this, but we're not like these individual age, demographics, phenotype, variant, multiple variants, all that. So we give examples of what's aggregate level of evidence versus when you consent, and we have developed a single-page consent form. And this is really just a data sharing consent. So if like a patient comes in and you order genetic testing as a physician or a genetic counselor, and you get a result back, and you simply want that data shared, and they're not consenting to a research study, they just want to share that data. You can use this one-page consent form for the patient to sign off that they're willing for their data to be shared. And that is what allows us to do this much more granular detail. And so in our ClinGen Genome Connect patient registry, the patients consent to this type of data. We then submit that into ClinVar, and then, because they filled out a very detailed phenotype form, we have all of these very detailed, specific phenotype features for a per-case basis that we can put into ClinVar because the patient has consented. And those of you who use ClinVar, you're like, oh, just tell me the phenotype. So sometimes you can put that in there if you go through this process. There's also other patient-level platforms where patients can share data in addition to Genome Connect. So the University of Washington group has a platform called MyGene2. And they have, last time I took this screenshot, 1,581 families who had posted their own or their child's phenotype, really hoping to get matches and get diagnoses. So they're putting their data out there. They can even upload their VCF files so that they can be downloaded by researchers and others to try to share. No consent is actually required because it's a platform where literally patients themselves upload their data and put everything in it, and the UW group is not doing it for them. So no consent is required at all. The virtue of uploading is their consent. Then they are actually participating in the matchmaker exchange, which is what I alluded to earlier, probably one of the first great examples of a truly federated network that supports rare disease analyses. So we launched this back in 2013, a group that had all these databases around the world, but none of them were connected. They were differently structured, and we all agreed we needed to figure out ways to share these data, but we're all in different countries. We couldn't send the data into one single platform. And so over time, we formed this federated network where all these databases are connected. We designed an application programming interface. And basically what's happening in these platforms is we call it genomic matchmaking, but basically you submit a case and you define one or more candidate genes. So you were looking in Seeker yesterday and trying to come up with your candidate gene for that case. Sometimes you come up with something and it's interesting, but it's not enough evidence to diagnose the patient. Well, you can take that candidate gene and you go stick it into Matchmaker Exchange along with the phenotype, and then hope to match some other patient who has at least one overlapping gene, and then look at the phenotype and see if it matches. And that process has been done now hundreds of thousands of times to actually discover causes of rare disease. The platform was designed so that when you enter your case, and if you match, there is dual notification of the submitters. The person who started the query gets notified, you got a match, but the person you matched against also gets emailed to say someone matched on you. I used to call this, I still do, data sharing for the paranoid. Because it's like, if someone is getting my data, I want to know about it. So you can put your data in, and it only gets discovered if somebody matches with you, and then jointly you can publish your paper together, and all is good. So we kind of designed that, in part in an era where people were a lot less likely to share data related to novel gene discovery. It's like the most coveted thing in the research rare disease world. And if you're close to discovering a gene, you don't want to put it out there and then someone scoops you. But we're like, yeah, but if you keep it held, you'll never match, and it just sits there forever. So it was like a way to kind of get people over that boundary line to say, well, put it out there in a protected way so that if there's another case, you'll match, you'll know each other how to match, and you can just work together and write that publication. It's been actually unbelievably successful. And we designed, when we first launched this platform, we designed what consent was required for this. And we actually defined that no additional consent was required in the clinical care scenario. Patient comes into a physician, they're expecting to get a diagnosis. So if you sequence them and find a candidate gene and you put it in Matchmaker, that's part of your job as a clinician to solve the cause of their disease. Also, research where discovering the cause of disease is part of the consent. No additional consent was required. The only time consent was required if you had unique or sensitive phenotype data that you were also uploading, or these were from research studies that were not consented for rare disease gene discovery or any sort of matching. Then you would have to get that additional consent. So to date, there's data from over 100,000 patients, from over 4,000 contributors from over 84 countries in this platform around the world. These are some of the matchmakers and the entries they have. It's had huge uptake around the world with almost 10,000 genes in the system. A lot of that is in Gene Matcher, which is sort of the more public-facing portal to use, whereas here within Seeker, we have the Matchbox system, but you can only use it if you're a user of Seeker. So like the public can't come out and get into Seeker and submit their gene. But at Gene Matcher, you just go in and you sign up for an account, and you can submit any gene you want. So most of the outside world uses Gene Matcher. So what we are doing today in Gene Matcher is what we call two-sided matching. Both individuals who submit each have a candidate. Like, I found this case and I found this candidate. I'm looking for a match with my candidate, and you match with somebody else who's a candidate. But occasionally, you've got a candidate, and maybe there's data out there, but whoever has it didn't identify that as a candidate, and you still want to match on their data. And so we're really starting to now think about how do we support one-sided matching and eventually what we call zero-sided matching. Nobody's got a candidate, but if the data's all brought together, you'll find it. So for this one-sided matching, we're now trying to launch other ways to think about this. So for example, sometimes you find a variant, and you're like, oh, this is an interesting variant, but you're not sure if it's a really good, you can't, in some ways you can't rule it out. And so you want to just go and find every case in the world that has this variant and know exactly what their phenotype is. You know, sometimes there's like one case in Nomad, and you're like, damn, I wish I knew what their phenotype was. Here, the Gene Matcher folks at Hopkins have launched Variant Matcher. So now you can go register very quickly and easily. You can enter a variant. If you put in your phenotype, then when they return results, they will give you the phenotype of the cases that they have. And so here's the list. So I put in this variant from one of the CMG cases, and these were the 8 cases that got returned, and they had all different phenotypes, or 4 of them were unaffected parents. So based on these 8 cases that got returned, we're actually able to quickly rule this out as a candidate variant, and therefore the gene itself. So this is a good way to then really, and they didn't select this as a candidate in their cases. Literally, it's going directly into the VCF files of these patients and saying it's in there, and this is the phenotype of that case. So this is really kind of one-sided matching. They had not considered this a candidate in their cases. We are now working, so that's one system where you can go directly to Variant Matcher. We're now working to do the same kind of building a federated network that we've done in gene-level matching, the Matchmaker Exchange, but doing this at the variant level. And these are some of the groups that are participating in a pilot that we're working on right now to try to get that federation connected. And these are just some of the screens where you can select which databases to search, Illumina's case log system, Cafe Variome, the Variant Matcher. So it's not live yet, but the group is actively working on getting this thing launched. And then once it's functional, I think more groups will then connect in through the API to participate. So that's where we are with that. So then, now that's kind of one side. You have to have some sort of hypothesis to search on. What about zero-sided matching? How do we just take all this raw data, it's unsolved for any disease, and really make heads or tails of it? So there's lots of raw data that's been generated from lots of projects. Since I've been involved in the Centers for Mendelian Genomics, I had put together this slide for a recent meeting where I was making the point that despite the fact that the 4 CMGs funded by NIH have generated data on 55,000 rare disease individuals, only 10% of that data has actually been effectively shared in dbGaP. And there's a long list of reasons for that, but a lot of it is just dbGaP has been totally overwhelmed with the ability to ingest this data, QC it, and manage it. And you can't submit a second data set until they've processed your first one. So, and then you can't submit a third until they've gotten through your second. So it took us over a year to get our first data set in there. So it's just a really difficult platform, and they recognize it. They recognize that they launched the system before we could sequence genomes, and it's now just totally overwhelmed. So we need different solutions. Good news is that NHGRI has funded a new platform called Anvil that will replace dbGaP as a system for depositing, storing, and accessing genomic data. And Anvil is a collection of different collaborating groups, partly centered here at the Broad, to really build this platform and support it. And the concept here is really changing from the world of dbGaP, where you had a central place that the data was going, and then for each group to analyze it, they had to apply for access. If they were granted it, they would then download the data into their servers and then do their analysis on it. So the new, go back. The new concept is to put the data in one place, although it could also be federated, but bring the users to the data, because trying to download genomes worth of data is very difficult. This makes it more accessible and secure. You can share tools in that ecosystem. It decreases the cost of storage and compute and facilitates collaboration, all sorts of good things. So you can load up both the data and collaborative tools all in one place and allow everyone access to both data and the tools. So the types of data, raw data, metadata. We're working on what phenotype data will be shared in Anvil, developing standardized ontologies. So the Broad CMG has now uploaded over a third of our rare disease data into Anvil. And you can see like individual level data, the phenotypes, whether they're affected, HPO terms, all of that, the collaborator it's from is all in there. And in fact, we've already had the first example where somebody else went into Anvil, took one of our cases and solved it using a new structural variant algorithm. So it's working already. And we're now working through the CMGs to define all of the rich metadata that will be attached on each case and agree on what is a standard set of data that should be there. Because just putting raw data is of no use. You don't know what the phenotype is and all the other things. This is just a list of the number of data sets that we are loading up into there. Now, this whole platform has been built on what is now called Terra, which was launched in beta mode just last month. You can actually go to terra.bio and you'll see this platform. And it's, as I mentioned, it's really helping with both storing the data, the tools, the analysis, and the results all in one place. And if you go, I was just on this this morning while I was putting this talk together, and you'll see the different data sets from 1,000 genomes, ENCODE, all the data that's in Nomad, Human Cell Atlas, the number of participants. You can browse that data. You can select the whole set or components of it that you want to do analysis on, all in there. And the same platform, in addition to Anvil, is powering the All of Us research workbench, the Human Cell Atlas project, the Cancer Institute's FireCloud platform, same actual infrastructure that's been built jointly between the Broad and Verily. So that brings me to All of Us. This is a lot of data we have to manage. There'll be data from 1 million or more people that is really needing to accelerate research and health, and it's being collected over several years and well-funded. And there's lots of data, participant-provided information, mobile health data, electronic health records, baseline physical exam, biospecimens, all collected in there. And the Broad, Verily, and Vanderbilt manage the Data Research Center, where all this data is coming together. It's in this, not like this traditional approach, but in fact, this come to the data approach that allows it to be much more secure and computable. So Vanderbilt is working on the part of getting the data out of electronic health records in standardized, structured ways and get it in. Verily is leading the sort of curated data repository and the common data model of all of this. And the Broad is leading the analysis platform to bring all the tools and the compute environment to this place. The All of Us data is consented for general research use and has no restrictions. That makes it, what that allows, instead of the traditional dbGaP, where for every individual project you have to individually apply, say, I want to do this with that. And then they review it, and they decide, and then they say yes or no. But then if you tomorrow think of a new idea, you got to come back and apply again. All right, now I want to do this. OK, we'll think about that, and we'll think about whether the data is allowed for that. So this is like completely general research use. So now we can approve the researcher, not the project. So you get like your passport and say, you're good to go. You can do anything you want within some reason. So that's how it's being structured. And they're tiering the data. There's some data that's public. Anybody can literally go on the platform and browse around and see cool data. Some of it is registered. You have to say who you are, and then your use and analyses are audited, trackable, and so we can see what you're doing. But it's you don't. It's pretty easy to get there. And then others, you have to specifically, it's more risky data that's sensitive, and you need to specifically say what you're going to do with it. So there's these three different things. To start, having an ERA Commons account. This is what all NIH investigators have to get a grant at NIH. But in the future, we will establish a procedure for citizen scientists, so literally anyone from anywhere, to be able to apply and get a passport to access data. So that is the ultimate goal. There are risks associated with that, to let anyone in the world get access to this data with, just kind of basic review. At the same time, the benefits of allowing anyone with a cool idea to analyze data has a lot of merit to it as well. So, you're here balancing those risks and benefits. To date, over 180,000 participants have enrolled in All of Us, 132,000 of which have completed all of the steps. So they've consented, they've gotten a specimen that's been submitted, et cetera. So this has been steadily increasing. What's amazing about this is that there's been a huge focus on correcting for what has been a largely white research population to date. And so there was a huge effort to recruit, actually more so than our US population in terms of underrepresented biomedical research and minority races. So to date, 80% of the participants are considered underrepresented in biomedical research. Now that's not just based on race, it's based on age, sex, sexual orientation, income, education, and geography or other factors that make people underrepresented. But 51% of it is specifically racial and ethnic diverse populations underrepresented. So this workbench, which is also on this Terra platform, is a place where you can go in, and it's like a cool little workbench, literally, where you can create a cohort. These are just me taking screenshots. You can select, the phenotypes that you want that are all coded in there in the system, and you can literally do like your research analyses and get your results on the platform, with not much effort, because the stuff is all there. It's really, really cool. Time is it? Okay, just one last quick topic. So recently I've been giving the whole question of thinking about data sharing in an era where you end up directly communicating with patients. In fact, that first two stories I told you, well, the second story, was around direct patient communication. And it brings up questions of not only responsibility, but liability. And in fact, that exact case that I showed you at the beginning of the talk, this is a slightly different version, which has the exact email that this woman sent me. And I was giving a talk up at the Jackson Labs last summer, and I got it was a CME credited course, you get all your reviews back, and I got this comment about, after sharing that case, non-clinicians dispensing recommendations via email made many of us very nervous, and likely legal counsel too. Clinicians are typically advised against this no matter how expert. I was like, ooh, yikes, what am I doing? So I actually did a little bit of online research trying to think about this topic of, well, what is my liability for having responded to this email? And so actually this is a really interesting paper that I found, thinking about this question back. Didn't work. And they, sort of unsolicited patient emails and giving tele-advice and dealing with all these stuff. They characterize encounters as type A and type B. Long story short, they sort of said, not only is it okay to do this in certain circumstances, but you actually could be considered to have a responsibility to do this. But you've got to do it in the right way. And I'm not going to go through all of the advice, but they say you don't want to make a specific diagnosis to this patient. You don't want to prescribe medicines. You're not their physician. But it's okay to provide general information, knowledge that might lead the patient to make the right decision, even though you're not making it for them. So I was like, okay, this is good advice. So then I went back to the emails. I was like, what did I say to this patient? And I described the case that we had seen the variant in, and that the variant was probably not causative in that case, general knowledge. I described what was in Nomad. I gave them our general variant interpretation that was then modified based on this data, and gave some basic information about the protein. And then I basically said, let me know when you get your husband's test, because she was then testing she and her husband for this variant. I'm guessing one of you will be positive. That further endorsed my assumption that this is probably a benign variant. So as I scrutinized my email, I'm like, I didn't say, therefore, you don't need to terminate your pregnancy, or any advice about how she should act in her case. And I didn't try to say, this variant is not causing anything in you. I didn't give her any specific diagnosis or advice about her care. I just shared knowledge. So that's an example, I think, of how to, be responsible and avoid liability and avoid taking on what should not be medical practice in essence. And the nice thing is the email ended with, is there a place we can go to document that he's not majorly affected so we can add to the science? These families want to contribute. So I gave them Genome Connect and told them to register there and put everything there. And then this was my favorite one. Just wanted to follow up on, baby was born, healthy, smiling, cooing. That helps you do your job every day, right? To get those sorts of things. Interesting that I'm just in the process of writing an ACMG session proposal on this whole topic. I've recruited Susan Wolf, Barbara Evans, and Adrian Thorogood, who are lawyers specializing in genetics, a patient advocate who has online platforms, to actually talk about this whole topic. Hopefully they'll accept my session proposal, and we'll have a great session at ACMG next year. But anyway, with that, I will just thank all of the funding and platforms and stuff that I talked about today, and I think we have a few minutes left for questions. Anyone, go ahead. Do the microphone, yeah.
Hi, about your federated data sharing platform. I just wanted to find out if there are any significant differences between sharing genome data as opposed to phenotype. Now, we try to share phenotype data, but this takes a number of days, maybe even weeks before we get a patient count of, we found so many at this particular hospital that satisfy this condition. Now, is that something that you deal with, or is that something, you have any advice on? Or
Sorry, I'm not quite sure I followed your question. The question is, are we getting phenotypic data for the cases?
No, there are I think there are two questions in here. A is that, do you support both searches for both genome and clinical phenotypes?
Oh, yes.
And the second part is, if you do support clinical phenotypes, what is the time frame? Do you do it better than like a few weeks? That's the second part of the question.
So in these platforms where you can come in and do analysis, you are able to search on the raw data, the sequence data, whether it's exomes, genomes, arrays, RNA-seq, whatever is there. And you might specify, only search data sets that have this type of data or something like that. But then you obviously also can search on, demographic factors. I only want to search on males under the age of 30 who are smokers and are of African American, whatever. And I want people with this clinical lab value or this diagnosis or this phenotypic feature based on HPO terms. So there's all different data types that you can search on, both genomic and phenotypic. The other thing, I still didn't quite follow your second question, but in terms of getting that phenotypic data, is that really what it?
So I think the context I'm asking from is that we work with distributed research networks called Shrine, which is known as ARCH, and then there's another one that we use called PubMedNet, and I work with a consortium of 16 other rare disease research, patient-driven research nodes. And what these operate on the principle that it's a central hub where someone puts in a request saying that we need someone with this demographic parameters and lab test results or what have you. And then it gets sent out to every node on that network. And then there's a data steward at each particular participating center. And then they come back in like maybe a week to make sure, and you only get patient counts. So they'll say, we found 20 people in Philadelphia, 15 in Cincinnati, what have you, right? Yep. And that's how it works. So I'm saying that, is your response time on when someone queries for phenotypic data at your distributed research network, is it faster? Is there anything different?
Okay, so now I follow your question. So for instance, in Matchmaker Exchange, it's real time. In fact, some of our analyst sessions are like, everybody agree we should submit it? Yeah, okay, do it right now, submit. And then it's like, oh, we got a hit. So like literally, that's real time because it's all API driven. No human has to say yes, release or anything like that. It's just as quick as electronic, communication. So for Matchmaker Exchange, that is done in real time, and we control it by virtue of the fact we just say both groups get notified, but it happens in real time. Same with Variant Matcher that I showed. We literally sat there, submitted, and bang, I got the email. back with the list of cases. So it's all real time. Same with the Anvil, Terra platforms. I mean, there's as you scale into large data sets, then there's compute time, right? But there's no gateway that somebody has to sign off on something.
Okay.
So it's fairly real time.
Thank you.
Yep. Other questions? I know it's getting late in the day.
To ask a little bit further about the exchange that you had with that particular patient, because
Yeah.
I think I fall closer to your perspective. Well, you're someone who posted that original variant up onto ClinVar. You technically are a canonical expert in this, and there are not a lot of people who you'd be able to go to, whether you're a patient or a provider. And you know, I'm wondering if you as a model of like consulting, like expert consulting geneticists for these things, like how do you advise like providers or clinicians to reach out about these types of cases as we're getting into the era where these things will be more automatically accessible?
Yeah, I mean, it's a great question, and you know, this whole, you know, most of the interaction, particularly around ClinVar data sharing, for example, is usually lab to lab, and you know, because I sequence a patient, I find a rare variant, I'm trying to interpret it, I see a couple other labs have submitted, but they just say, you know, VUS, and there's not much information there. Maybe there's a code, PM2 or something. But I kind of want to know, who'd you see that in? What are the details? And so there is, you know, communication that's happening largely through email between laboratories. And there's kind of this, at least from my perspective, because I still get all the emails for the LMM. You know, I would say we get an email once every couple weeks based on having shared 18,000 variants into ClinVar. And it's usually some lab asking my opinion on a variant we put in. Maybe they want a little more information. It is extra work because, you know, either I forward it on to someone in the lab who then digs up the case and sends it back and say, we saw it in a patient with X, Y, and Z. But I would kind of, you know, if you look holistically at ClinVar, I would say the initial interpretation of most variants, we save time because we go into ClinVar and somebody's already read the literature, collected it together, and we're more like kind of updating. And so we've saved time by baseline data being there. But we've then lost time because we're now communicating with each other. Why did you say that? I disagree with that. And can you share me your data? So like net, I feel like we're kind of about the same level of resources, but although potentially even reduced. But there is this like responsibility to some extent. And so I think we all kind of, you know, reach out when there's a really critical case, like this woman's pregnant and they're considering termination. Whereas other cases where I'm like, it's a VUS, it's one of the, you know, 100th VUSs we saw that week. I don't reach out to labs on every VUS, right? So we all kind of wait for the cases that are where we really think this could make a difference, and it's kind of a professional code of whatever. But you know, the patients, it's their case. Every case is important. So you know, trying to figure out as patients become more and more aware of ClinVar, and they're going to send the email every single time. And so it's a great question as to how do we scale this world. And you know, I would argue we want to increasingly structure our data and allow tiered access to it in ways that, you know, someone could, through like the All of Us research with a data passport, have, you know, I have this variant and I want to know what the patients that have it are. Can I get access to this data? And I don't have to go dig it up out of my database and send an email with it, right? So I think, I don't know if it's quite getting at your question, but you know, I think this is a complex world as to how we think about our responsibilities, but the workload requirements of it, and as well as our liabilities.
Thanks.
Yeah. Any other last questions? Do we have Sonia yet? What's that? Are we going to move on? Yep. Okay. We have only 3 minutes left, but I think we can start early. All right, well, thank you all for that, and turn it over.